Okay, so what does it really mean to be human right? Like, is it some spiritual thing? Is it
Yeah.
because we can reason? Or is it something deeper, something actually like written into us?
Well, early on in the whole Human Genome Project thing, Renato Del Becco, he said that the sequence of human DNA is the reality of our species.
Oh, wow.
Yeah, and Walter Gilbert, he even said that figuring out the entire human genome sequence, it was like going after the Holy Grail.
Really?
Yeah.
It really makes you think, about how we see ourselves.
It's a huge shift in thinking, right?
Uh,
Like for centuries, philosophers have been trying to figure out humanity, but mostly through abstract ideas. And
right.
now science is saying, hold on, we have a tangible thing right here, our DNA sequence.
Yeah.
So can we really just boil down what a species is to its biological code? I mean, is that all there is to it?
That's a great question.
Yeah.
And it leads us perfectly into today's deep dive, the Human Genome Project. This massive effort to map and sequence the whole human genome.
Huge undertaking, right?
I mean, it officially started in 1990,
Uh-huh.
and they said it was done in 2003. And it wasn't just one country doing it. It was a global team.
Hmm.
The U.S., the U.K., Japan, France, Germany.
China?
Everyone was involved.
Exactly. And the researchers, they knew how big this was. They compared it to landing on the moon or splitting the atom.
Yeah.
Like they were ushering in a new era of the genome, they said.
It's kind of mind-blowing when you think about the philosophical implications of that kind of statement.
No kidding. So we're going to go deep into this project today.
Deep dive.
Yeah, exactly. A deep dive into how this whole thing even got started, how they actually did the mapping and sequencing, the whole genomics part of it, and then what's happened since the post-genomic stuff.
Right.
But we're not going to just rehash the science. We're going to look at the big philosophical questions that came out of it, especially the stuff, that philosophers would be interested in about existence and knowledge.
Absolutely, because beyond the scientific achievements themselves, right, what did this project do to how we understand life and what it means to be a person? Like, did it change our fundamental concepts?
That's what we're going to unpack today.
Buckle up. Exactly.
So first off, how did this whole HGP thing even come about? It's not like someone just woke up one day and said, hey, let's sequence the entire human genome.
No, not quite. It actually had its roots in earlier ideas about heredity, like the really basic stuff.
Right.
So the seeds were planted in the mid-1980s.
Mid-'80s, okay.
Yeah, with guys like Robert Sencheimer, Renato Del Becco, Charles DeLisi. They started putting forward this ambitious idea.
Okay.
And it did get some support from some big names in molecular biology and human genetics.
Oh.
But it wasn't like everyone was immediately on board.
Oh, really?
No, you have to think about how biological research was done back then.
Okay.
So a lot of molecular biologists, they were worried about this massive sequencing project.
Oh.
That they called junk DNA back then?
Oh, yeah.
They thought it would be a huge waste of resources.
Really?
Yeah, and they were afraid it would suck up all the funding, leaving less money for smaller, more focused projects.
That makes sense.
And there was also this concern that just having the sequence, the raw data, wouldn't be enough.
Like, what does it all mean? How do these genes work together? That was a whole other question. It's like having this huge book, but you can't read the language. It's just letters on a page.
You have the pieces, but you don't know how they fit together.
Exactly. So to try and address these concerns, they did a bunch of feasibility studies and reports. Like the Office for Technology Assessment did one called Mapping Our Genes. And the National Research Council, they did mapping and sequencing the human genome.
Okay.
And these reports tried to reassure the people who were skeptical.
Okay.
They said, hey, this isn't just one giant project. It's a bunch of genome projects.
Oh, okay.
And they said it wouldn't necessarily be on the same scale as those other huge projects like the Manhattan Project or the Apollo program.
Okay.
And they also stressed that studying the genomes of other organisms would be crucial, to really understand our own.
Right, so comparing ourselves to other species.
Exactly. It's an approach that makes a lot of sense in biology, even from a philosophical standpoint.
So it's like recognizing that we're all connected in some way.
Yeah, and that to understand ourselves, we need to look at the bigger picture, like the whole web of life.
Okay, that makes sense.
Yeah.
So did these reports work? Did they convince the skeptics?
Well enough to get the ball rolling.
Okay.
The Department of Energy, they actually made the first big push in 1986.
The Department of Energy.
Yeah, which kind of ruffled some feathers at the National Institutes of Health. They were the ones who usually funded this kind of biomedical research.
Oh, I see.
But eventually, the head of the NIH, he came around and gave his support in 1987.
Okay.
So by 1988, both agencies had funding from Congress. The National Research Council, they estimated it would cost around $3 billion.
$3 billion. Wow.
Big science, right?
Yeah, no kidding. And it wasn't just the U.S.
No, it quickly became a global effort.
Okay.
I mean, France, the UK, Japan, Germany, China, they all joined in.
So everyone recognized how important this was.
Yeah, I think there was this shared understanding that this knowledge about human biology, it was important for all of humankind.
Right. It wasn't just about one country or one group of people.
Exactly. And we saw some major contributions from these international partners early on. Like in France, they had CEPH and Genethon. They were essential for the genetic mapping part.
Okay.
And then in the UK, they had the Sanger Center. That became a major hub for sequencing.
Okay.
So it really was a global team
A
effort.
true collaboration.
Absolutely.
So they had the funding, they had the team, but where did they start? I mean, did they just immediately jump into sequencing 3 billion base pairs?
Well, they had a more strategic approach.
Okay.
Map first, sequence later. That was their motto.
Map first, sequence later. Okay.
Yeah, it was a practical way to deal with this incredibly complex problem.
So what did that involve?
Well, the early years focused on creating maps.
Maps. Yep.
Yeah, two kinds, actually, genetic maps and physical maps.
So what's the difference?
Well, genetic maps, they show the order of genetic markers on chromosomes based on how often they recombine.
Okay.
And physical maps, they arrange cloned DNA fragments in order.
So they're like creating a framework, a scaffold, before they start filling in all the details. Yeah.
Exactly, like writing an outline before you write the whole book.
Okay, that makes sense.
Uh-huh.
And what are those other organisms you mentioned earlier? The model organisms.
Right. Those were important, too. They were running parallel with the mapping efforts. They were using them as testing grounds to figure out the techniques and to get insights into basic biological processes.
So they were kind of like practice runs for the human genome.
Yeah, exactly.
Mm-hmm.
And they did make a lot of progress with the mapping, Genethon in France. They created some really detailed genetic maps.
Okay.
And by 1995, they published a pretty complete physical map of the human genome.
Wow. So they had a roadmap.
Yeah, they were ready to start sequencing. But that turned out to be a bigger challenge than they expected.
Really?
Yeah, even with all the work at places like the Sanger Center and the ones funded by the NHGRI.
The what?
Oh, sorry, the National Human Genome Research Institute.
Okay.
They were making progress, but the costs were still really high and they weren't getting the results they wanted as quickly as they wanted. So they managed to sequence the genomes of some smaller organisms.
Okay.
But the human genome, it was still this massive obstacle.
So they had a roadmap, but they were stuck.
Kind of, yeah. But then things got a little more interesting.
Oh, what happened?
Well, in 1998, this guy, J. Craig Venter, he was at Tiger at the time.
Okay.
He announced that he was partnering with Applied Biosystems to sequence the entire human genome.
The whole thing.
Yeah, the whole thing. And he said they could do it in just 3 years.
Wow.
And for a fraction of what the HGP was spending.
Really?
Yeah, they formed this company called Celera Genomics.
So a private company was now competing with the public effort.
Exactly. And it was a big deal. It really shook things up in the scientific community.
I bet. So how were they planning to do it so much faster and cheaper?
Well, they were using a different approach. It was called whole genome shotgun sequencing.
Whole genome shotgun. Okay, I've definitely never heard of that.
Yeah, it was pretty radical. Basically, they were going to break the entire genome into millions of tiny pieces, sequence them all, and then use supercomputers and fancy algorithms to put them back together based on overlapping sequences.
So it's like taking a puzzle, scattering all the pieces, and then trying to put it back together without the picture on the box.
Yeah, kind of.
Right.
And the HGP scientists, they were pretty skeptical.
Oh, really?
Yeah, they were worried about all the repetitive DNA in the human genome.
Yeah.
They thought those repeating sequences could cause errors when they tried to put everything back together.
That makes sense.
But Venter, he had already used this method to sequence the genome of a bacterium.
Okay.
So it had worked before, but the human genome was way more complex.
Right.
So it was a gamble.
So what happened? Did the HGP just give up?
No, they felt the pressure, this race to the genome, as the media called it.
I bet.
They got more funding, and they sped up their timeline. They wanted to have a working draft by 2001. They even changed their strategy a little bit. They started using larger DNA fragments to help with the assembly process.
So they were adapting to the competition.
Exactly. And they had some successes along the way, like they managed to sequence the genome of C. elegans, the roundworm.
A worm?
Yeah, it was a big deal, though. It showed that their approach could work for a complex animal.
Okay.
And then came the big announcement, June 2000 at the White House.
Oh, wow.
Clinton was there, Venter Collins, everyone.
So they declared victory.
They said the human genome had been sequenced.
So the race was over.
Well, not really. It was more like a truce.
A truce.
Yeah, neither sequence was actually complete or totally accurate.
Oh, so it was more of a symbolic moment?
Yeah, a moment of shared celebration. Maybe a little premature.
I see.
And the details, they didn't come out until much later. The actual scientific publications with all the methods and findings, they weren't published until February 2001.
So almost 8 months later.
Yeah, the HGP published in Nature and Celera published in Science. And even then, those publications, they made it clear that these were still drafts.
Oh, really?
Yeah, the HGP draft, it covered about 94% of the genome, but there were gaps and some parts were less accurate.
Okay.
And it's important to note that Celera, they actually used a lot of the HGP's data to put their sequence together.
Really?
Yeah, the HGP was making their data publicly available as they went along.
Okay.
So it raises the question of whether Celera could have done it on their own with just their whole genome shotgun approach.
Right, because the human genome is so big and repetitive.
Exactly.
So it was a bit of a team effort, even though they were competing.
It's a great example of how public and private science can interact in complex ways.
So when was the project officially finished?
Well, they declared it complete in April 2003.
2003, okay.
Yeah, a bit earlier than they initially thought, and it actually came in under budget.
Wow.
They had a much more complete and accurate reference sequence,
Yeah.
and they made it publicly available.
So a big win for open science.
Absolutely. It's been a foundational resource for so much research since then.
But it still wasn't 100% complete.
No, not entirely. There were still some tricky parts, mostly in those highly repetitive regions.
Okay.
But it was a huge accomplishment.
So the project was done, the genome was sequenced. What happened next?
Well, there were all these promises, right?
Oh, yeah?
About revolutionizing medicine, understanding all the causes of diseases.
Right, like a cure for everything.
Yeah, there was a lot of hype.
So did it live up to the hype?
Well, that's where things get a little more complicated. The public, they were really excited about the HGP. And that excitement, it was fueled by these really optimistic promises.
Right.
Some people were saying that we'd be able to cure all diseases, prevent them even.
Wow.
Yeah, like Daniel Koshland, he even suggested that genes were responsible for things like poverty and homelessness.
Oh, wow.
Yeah, it was a pretty bold statement.
No kidding. So were they right? Did all these medical breakthroughs happen?
Well, not exactly. I mean, there have been some amazing advances.
Okay.
Especially in our understanding of single gene disorders. Like the number of known genetic causes for Mendelian diseases has gone way up.
So those are diseases caused by a single gene.
Exactly. And that's led to better diagnosis and even some targeted therapies.
Okay, that's great.
But for the more common complex diseases, the ones that affect most people, it's been a lot harder.
Nice.
Like heart disease, diabetes, cancer, those are all influenced by multiple genes and the environment.
Right, it's not just one gene, one disease.
Exactly.
Yeah.
And figuring out all those interactions, it's really difficult.
So the initial hype was a bit overblown.
Yeah, you could say that.
But surely the HGP had some positive impacts.
Oh, absolutely. I mean, it might not have led to all the cures they were hoping for,
Right.
but it was a huge boost for basic science.
Okay, how so?
Well, it led to the development of incredible new technologies for mapping and sequencing DNA.
Okay.
And they had to come up with all these new bioinformatics tools to handle these massive data sets.
There's a technological revolution?
Yeah, basically. And those technologies, they've had a huge impact on all of the life sciences, not just human biology.
Oh, wow.
And they made some pretty surprising discoveries along the way.
Like what?
Well, for one thing, they found that humans have way fewer genes than they thought.
Really?
Yeah, they were expecting something like 100,000 protein coding genes.
Okay.
But it turns out we only have around 20,000.
Wow, that's a lot less.
Yeah, it was a big surprise. And it means that our complexity, it can't just be explained by the number of genes we have.
Okay, so what else did they find?
Well, they learned that all that non-coding DNA, the stuff they used to call junk DNA, it actually plays a really important role.
Really?
Yeah, it's involved in regulating gene expression.
So it's not just junk.
No, not at all. It's essential.
So the genome is way more complex than they initially thought.
Yeah, it's not just a simple blueprint.
Yeah.
It's a dynamic, interactive system.
And that complexity has made it harder to find those simple gene-disease links.
Absolutely.
Okay, so where are we now?
Yeah.
What's the current state of genomics research?
Well, sequencing technology has gotten insanely good.
Yeah.
Like it's now possible to sequence a whole genome for less than $1,000.
Wow.
And that's led to the rise of personal genome projects.
So people can get their own DNA sequenced.
Exactly.
That's pretty cool.
And it's also leading to new approaches to medicine, like precision medicine.
Okay, so what is that?
Well, it's the idea of tailoring treatments to an individual's specific genetic makeup.
So, like personalized medicine.
Exactly.
Okay.
And there are these huge biobanks now that are collecting genetic and health data from millions of people.
So they're trying to understand the links between genes and disease on a massive scale.
Exactly.
Wow, it's like we've gone from looking at individual genes to studying whole genomes and how they interact with the environment.
Yeah, it's a whole new way of thinking about biology and medicine.
So are we in a genomic utopia now?
Well, not quite. I mean, we've made a lot of progress, but there's still challenges.
Okay.
We have all this data now, but figuring out what it all means, that's still a huge task.
Right? It's like having a giant library but not knowing which books to read.
Yeah, Jenny Reardon, she called this the post-genomic condition.
Okay.
We have this huge amount of data, but we're still struggling to make sense of it all.
So the real challenge now is interpretation.
Absolutely.
So beyond the science, what about the ethical and philosophical implications of all this? I mean, sequencing the human genome, it's got to raise some big questions.
Oh, absolutely. I mean, right from the beginning, there was this recognition that the HGP would have huge ethical, legal, and social implications.
Okay.
And it wasn't just an afterthought.
Oh, really?
No, James Watson himself, he was the first head of the National Center for Human Genome Research.
Okay.
And at the press conference announcing his appointment, he said they'd set aside some of the funding to study these issues.
So they were thinking about the ethical implications right from the start.
Yeah, which was pretty forward-thinking for a big science project like this.
And how much did they set aside?
Initially, it was just 3%.
3%.
But
Right.
it led to the creation of this group called ELSI. Yeah, it stands for Ethical, Legal, and Social Implications.
Okay.
And it became a pretty big part of the HGP.
So what did they do?
Well, they funded all sorts of research into things like genetic testing, genetic screening, privacy, confidentiality, the potential for misuse of genetic information.
So they were trying to anticipate the potential problems.
Exactly.
And who was involved in ELSI?
It was a mix of people, philosophers, ethicists, legal scholars, social scientists.
Wow, so they brought in a lot of different perspectives.
Yeah, they recognized that these issues, they were too big for just scientists to handle. It needed a broader conversation.
Absolutely.
Right.
So beyond those more practical ethical concerns, were there any big philosophical debates that came out of the HGP?
Oh, yeah, definitely. Like
Yeah.
some really fundamental questions about what it means to be human.
Okay, like what?
Well, remember those quotes we talked about at the beginning?
About the human DNA sequence being the reality of our species.
Exactly. Some people were saying that the genome held the key to understanding our very essence. And this idea of geneticization, it became really powerful.
Geneticization.
Yeah, it's the tendency to explain everything about us through our genes,
Oh, I see.
like our physical traits, our behaviors, even our social problems.
So it's like saying our genes determine who we are.
Yeah, and that idea, it was used to justify the massive investment in the HGP.
Okay.
But philosophers, they raised some serious concerns about this.
Like what?
Well, for one thing, it's not clear that genes are the only thing that matters.
Where the environment plays a role, too.
Exactly.
Yeah.
And our experiences, our choices.
So it's not all predetermined by our genes.
No, not at all. And philosophers, they also question this idea of genetic reductionism.
Okay, what is that?
Well, it's the belief that everything about an organism can be explained by its genes.
So it's like saying we're nothing more than our DNA.
Yeah, basically.
But that doesn't seem quite right.
No, and philosophers pointed out that things like health and disease, they're not just about genes.
Very good.
They're also about how an organism functions in its environment. And they're also about our values as a society.
So like what we consider normal or abnormal.
Exactly.
Okay, that makes sense.
And then there's genetic determinism.
Okay, what's that?
It's the idea that our genes control our destiny.
So like we're just puppets of our DNA.
Yeah, that's a pretty extreme version of it.
Yeah.
But the idea is that our genes have this powerful, even inevitable influence on who we are and what we become.
Is that true?
Well, it's a lot more complicated than that.
Oh, yeah.
Even for single gene disorders, the ones that are most clearly linked to genes, there's still variability.
Okay.
Like, not everyone with a particular gene will develop the disease.
Oh, I see.
And even if they do, the severity can vary.
So there are other factors at play.
Exactly.
Mhm.
And for the complex diseases, the ones that are influenced by multiple genes and the environment, it's even more complicated.
Right.
Trying to separate the effects of genes and environment, it's almost impossible.
Okay, so it's not nature versus nurture.
No, it's nature and nurture working together.
Right.
They're intertwined. And trying to say which one is more important, it's kind of pointless.
So these ideas of geneticization, reductionism, determinism, they're all oversimplifications.
Yeah, they're tempting because they offer this simple explanation,
Right.
but biology is messy. It's complex.
But didn't these ideas help to promote the HGP?
Oh, yeah, definitely. They were part of the sales pitch.
Okay.
Like Walter Gilbert, he said that one day we'd be able to carry around a CD-ROM with our entire genome on it.
Really?
Yeah, and he said we'd be able to say, here is a human being, it's me.
Wow, that's a pretty reductionist view.
Yeah, but it was a compelling image.
I guess so. And did people buy it?
Well enough to get the project funded.
So the hype worked.
You could say that, but it also created some unrealistic expectations.
Right, like about all those cures that haven't materialized.
Exactly. And some people, they're worried that this focus on genes, it's distracting us from other important factors.
Much what?
Well, like social and environmental factors,
Yeah.
like poverty, access to healthcare exposure, to toxins.
It's not all about our genes.
No, definitely not. And focusing too much on genes, it can make us think that we're not responsible for our health.
Right, like it's all predetermined.
Exactly. And that can be dangerous.
So the HGP, it's had a huge impact on science and medicine.
Absolutely.
But it's also raised some really big philosophical questions that we're still grappling with.
Yeah, and those questions, they're not going away anytime soon.
Okay, so what's one of the biggest questions that came out of the HGP?
Well, the whole idea of sequencing the human genome, that raises some interesting questions.
Okay, like what?
Well, whose genome are we talking about?
Right, there are billions of people on Earth.
Exactly. And everyone's genome is different.
So how do you choose one genome to represent everyone?
Yeah, that's a big problem.
Yeah.
And it turns out they didn't actually sequence one person's genome.
Oh, really?
No, they used DNA from multiple people.
Oh, okay.
They created a composite sequence.
So it's like a Frankenstein genome?
Kind of. And for most of the initial reference sequence, they used DNA from about 20 people. And a lot of it might have come from just one or a few individuals.
Oh, wow.
So it raises this question of how representative can this composite sequence be?
Right, it's just a small sample of all the genetic diversity out there.
Exactly. And early critics, they pointed out that a lot of the variations in our DNA, they don't actually have any effect on our traits.
Okay.
So they're functionally equivalent.
So why sequence them?
Well, at the time, they didn't know which variations were important and which ones weren't.
Oh, I see.
And they were also worried that the reference sequence, it might contain some hidden defects.
Like mutations.
Yeah, mutations from the people whose DNA was used.
Okay.
And from an evolutionary perspective, mutations, they're not always bad.
Right, they're the sorts of variation.
Exactly. And variation is what allows species to adapt and evolve.
So it's not always about having a perfect genome.
No, definitely not.
But there's still this idea out there that there's a right way to be genetically.
Yeah, it's a slippery slope.
Like this idea of genetic normality.
Exactly. And the reference sequence, it kind of reinforces that idea.
So it's like saying, this is the ideal human genome.
Yeah, and any deviation from that is a problem.
But that's not how it works.
No, every genome is unique.
And diversity is good.
Absolutely.
So what are they doing now to address these concerns?
Well, they're constantly updating the reference genome. They're adding more sequences from more diverse populations.
So trying to make it more representative.
Exactly. And they're also exploring new ways of representing the human genome.
Okay.
Like instead of having one linear sequence, they're looking at ways to show all the variation.
So like a 3D map of the genome?
Yeah, something like that.
Yeah, that's pretty cool.
And they're also trying to move away from this idea of a single reference sequence.
Okay.
Like they're developing these things called pangenomes.
Pan-genomes, okay.
Yeah, which are basically collections of genomes from many different individuals.
So it's like a database of human genetic diversity.
Exactly.
So they're trying to move away from this idea of a single normal genome.
Yeah, they're recognizing that diversity is the norm.
And it's a good thing.
Absolutely.
So the HGP, it's raised all these big questions about what it means to be human.
Yeah, and those questions, they're not going away anytime soon.
So where do we go from here?
Well, I think we need to keep having these conversations about the ethics of genomics, about what it means to be human in this new era of genetic knowledge.
Right, because it's not just about science.
No, it's about our values as a society.
Exactly. So it's up to all of us to figure out how to use this knowledge responsibly.
And wisely.